Hello and welcome to Nikolai's genetics lessons, and today's problem is alkaptonuria is an autosomal recessive disorder that is associated with the production of brown sweat and dark brown urine. In a certain population, approximately 1 out of 90,000 people are affected. What is the approximate carrier frequency for this condition? You easily can solve this problem if you know Hardy-Weinberg formula, which tells us that frequency of the three genotypes, which is made by two alleles in a population, would equal to p squared plus 2pq plus q squared, and all three genotypes equal to 1. So we have here in this population only two alleles, p, which is going to be dominant or normal allele, and allele q, which is going to be mutated allele that in homozygous form cause this genetic disorder. For example, maybe you get used to different letters such as dominant allele A and recessive allele A. Then p squared would stand for the genotype which is homozygous dominant, 2pq would stand for the genotype which is heterozygous, and q squared stand for the genotype which is homozygous recessive. And according to our problem, 1 out of 90,000 people would belong to this genotype, so would have this genetic disorder. So that means they would belong to this genotype and phenotype, and these two genotypes would make another phenotype, because this is autosomal recessive genetic disorder. So people who are heterozygous would show the same phenotype as people who are homozygous dominant. If we would be able to find the frequency of the dominant allele and recessive allele in this population, we would be able to solve this problem. Take a look. Homozygous recessive genotype, which is q squared, or A times A, we can say A times A. Frequency of this genotype and phenotype equal to 1 out of 90,000. So we can say in this case that q squared equal to 1 out of 90,000, and in order to find the frequency of the recessive allele, or q, we just have to take a square root from this number. So in order to find q, we just have to take a square root. So now we know that the frequency of the q would be 1 out of 300. Now, if we know frequency of the recessive allele, which is 1 out of 300, we instantly know the frequency of the dominant allele, which is going to be 299 out of 300. So if we add these two frequencies, we are going to get 1 or 100%, because this gene pool consists of only these two alleles. Now let's read our question again. What is the approximate carrier frequency for this condition? So we are looking for a frequency of the carriers. This part of the formula stands for the frequency of the heterozygous genotype, or frequency of the carriers, 2pq. And this equal to heterozygous genotype. Now we know frequency of the dominant allele, which is 299 over 300, and we have to multiply by frequency of the recessive allele, which is 1 over 300. So what we are going to get here, 299 divided by 300, we can say without even calculator that this is approximately going to be 1. So basically we divide the same number but the same number. So here we basically have 1, and 2 times 1 is going to be 2 divided by 300. So the answer is going to be approximately 1 out of 150. And we can change the sign also to show that this is just approximation. This is not exact number, because this is just a frequency, but we expect that number of the heterozygous people or carriers are going to be 1 out of 150. And this is answer B. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. and see you in the next video. Goodbye.